Control of bacteria. Now we have a question that why control of microbes or bacteria is necessary. So as we know that bacteria cause number of diseases in humans and they cause damage to plants or agricultural industry. So we have to control these microbes ताकि इसके adverse effect से बचा जाए या control किया जाए इन्हें. For the control of bacteria, we have two basic methods. Number one is physical method and number two is chemical method. So the process in which we use physical agents to control bacteria or other microorganisms, it is known as sterilization process. The various physical methods of control are dry heat, moist heat, gas, filtration and radiation. We will look into it one by one. Talking about the dry heat, it actually dehydrates the cell, and as a result of dehydration the cells die. And the other one is that oxidation of chemical constituents of microbes, and as a result of oxidation the object or microbe is converted into ashes. The example of dry heat are the common ovens used in laboratory. In our bio lab we used a dry heat oven for sterilization of various instruments or products, and the other example is incinerator shown in herein, and they are used in hospital for the cleansing or sterilization of hospital waste. Moist heat actually causes coagulation or thickening of protein, और जिससे protein functioning halt हो जाती है and it eventually kills the microbe. Talking about the types of moist heat, firstly we have steam under pressure. We know the example of autoclave, or most common example is a pressure cooker used at home. The basic principle is that the boiling point of water is usually increased inside the pressure cooker or autoclave, और जिसकी वजह से temperature of steam भी increase हो जाता है. In autoclave operation we increase the temperature of water to 121 degrees centigrade. It is actually the point at which the water boils, or it is not the pressure which kills the bacteria, but it is the increased temperature which kills the bacteria. For some other objects we have another method which is non-pressurized steam. इसमें steam तो होती है for killing of microbes, लेकिन इसमें it is not covered or it is not pressurized actually. So इसमें एक अलग container होता है जिसमें steam generate की जाती है, and in another container we have object to be sterilized, और ये heat sensitive objects के लिए है, and the object is subjected to this steam for period of 30 to 60 minutes, and it results in sterilization of the object. And then we know another process which is pasteurization. It is actually employed for the sterilization of liquids mostly. For example, we can gauge that milk is usually sterilized by the process of pasteurization. इसमें एक specific temperature पर specific time duration के लिए ऑब्जेक्ट को सब्जेक्ट किया जाता है, और जिसकी वजह से we get the control of bacteria or sterilization of bacteria. The other one is boiling water, and it is most common, लेकिन it is not reliable. We usually employ it at home for water and we use boiling of water to make it fit for drinking. The other physical method is that of radiations, and radiations below 300 nanometer are usually used to control the bacteria. For example, UV radiations. In addition, gamma rays are also used to control bacteria. We have another technique which is called filtration. For example, in membrane filtration, जो बहुत sensitive materials होते हैं like hormones or vitamins, उनकी sterilization के लिए we use this filtration process. इसमें we have filter जिसमें बहुत small pores होते हैं through which bacteria cannot pass, while fluid can pass easily. So इसकी sterilization easily हो जाती है. And then we have another method, which is that of use of gases. For example, we have ethylene oxide, and it is actually used for, to sterilize object which can't sustain high temperature or temperature above 60 degrees centigrade. So plastics, optics, and electrics are usually sterilized by the gases.